Imagine you're the guardian of a village. Recently, the village is being raided by monsters from a nearby mountain, so you and your fellow guardians decide to bombard the mountain with all types of weapons. It was rains of fireballs and lightning strikes and magic spells, and for some time, it did seem that peace is restored to the village. But it wasn't long before those monsters began terrorizing the village again, so you decide to go to the mountain and investigate. And what you find are giant beasts who are somehow completely unfazed by your attack. And to your terror, you saw one giant beast shapeshift into hundreds of little monsters who are also immune to your attacks and are now ready to mount their own defense. That's enough fantasy for now, but what sounds like a horror fiction is actually a very real challenge that oncologists are facing. When a cancer relapses, it means that the treatment did not fully eradicate all cancer cells, and after some time, tumors will form in the patient's body again. My research topic, polyploid giant cancer cells, or PGCCs, have been gaining a lot of interest because of their pivotal role in cancer treatment resistance and relapse. PGCCs are a very unique population of cells. As you can see, they are huge, they have distinct physical properties. They can survive thousands of folds of treatments compared to normal cancer cells, including chemotherapy and radiation therapy. And worst of all, every PGCC has the ability to divide into hundreds of little cancer cells that are also resistant to treatment and can now form tumors that are more difficult to kill. So, every single PGCC that evades treatment has the potential of causing cancer relapse. And also, that story at the beginning was not at all far-fetched. It is actually what's happening in the bodies of many, many cancer patients. But fortunately, there is a silver lining that every survival advantage comes with a cost. Giant beasts like these have vulnerabilities. When you think of such a giant, you think of the extensive physical structures they require to even support themselves, as well as the enormous amounts of nutrient they must consume to sustain everything going on in their cell body. And those two simple ideas, structure and energy, are actually part of what drove my research topics. I studied the cytoskeleton structure and energy metabolism of PGCCs. Cytoskeletons are structural proteins, think bones and muscles of the cell. Cancer cells metastasize to other parts of the body and initiate new tumors, and PGCCs are some of the most migratory and invasive cancer cells. As they migrate through solid organs, fatty tissues, bloodstreams, open spaces, narrow passages, cytoskeletons must support their structure or deformation in all of these different physical microenvironments. Metastasis would also be more successful if a cell is able to move in a particular direction in a more persistent manner. Cytoskeletons support this process by aggregating at one end of the cell to reinforce that direction of migration, as well as by organizing the distribution of smaller functional units called organelles associated with energy metabolism to concentrate energy production to the direction of migration. And speaking of metabolism organelles, you're all familiar with one of them already, the mitochondria, commonly known as the powerhouse of the cell. The mitochondria can process various forms of nutrients into energy, including our lab's focus, fats, or more scientifically, lipids. I found that PGCCs can store a lot of lipids in their cell body in the form of hundreds of lipid droplets, whereas normal cancer cells have only a few lipid droplets. My data show that PGCCs rely much more heavily on processing these lipids in their mitochondria to produce energy. So I used chemicals that target the lipid metabolism process in conjunction with chemotherapeutics, and indeed, this combination was much more effective at killing PGCCs than either drug by itself. Through studying the fundamental biology of these giant cancer cells, my research identifies vulnerabilities to exploit. Laboratory results about lipid metabolism can eventually go to the clinics, where commercially available drugs with similar effects against lipid metabolism can maybe be used to improve the efficacy of traditional chemotherapeutics. Ultimately, we are all guardians. We, the scholarly ones, study our enemies tirelessly in the hopes of giving our cancer patients a brighter tomorrow. Thank you.